Hi, this is Dr. Debdutta Bandyopadhyay speaking. By education and by profession, I am a molecular biologist and cell biologist. Presently, I am working in a pharmaceutical company in India. Since past couple of weeks, I have been posting different videos on various aspects of coronavirus, and recently I got some requests from my friends to throw some lights on coronavirus vaccines. Please keep in mind the target audience for this video is common people who are not from biology, from the field of biology. So I will make it as simple as possible and to give some idea about what are the coronavirus vaccines, how does it work, and what are the progress going on regarding this vaccine field across the globe. Just a snapshot, about 90 different vaccines are being developed against SARS-CoV-2 by different teams and universities across the world. Now, some of these groups, they are using some very new way to develop the vaccine which are not really licensed before, and they have already started some human trials. When we say injecting formulation into volunteers, means injecting the vaccine into human. All the information that I am giving over here, most of them were collected from a publication in the one of the most famous journal in biology, Nature, volume 580, which was published in 30th April. So that is about 3-4 days back. So let's start with the first thing is how does immunity work? To get information about vaccine, the first thing we need to know what vaccine is, how vaccine does work, and to know that we need to know how does our body's immunity work. So that is the first thing. Let's start from a coronavirus infection. So this is a coronavirus with its spikes on the outside and a small RNA inside which acts as their genome, just like we have DNA, 23 pairs of chromosome. You must have heard about it. But coronavirus have only one single-stranded RNA, which is about 30,000 bases long. That means those A, G, C, T, which makes the DNA. Here A, G, C and U, they are all spread around this thing, and there are about 30,000 of them. When the virus enters the body through this ACE receptor, by the way, I have discussed this pathway in my first video in this series, where I have discussed different medicines that are being trialed, being developed for the coronavirus infection. You may go back to my YouTube site and check that. Now, as it gets entry into the host cell, that is our cell, it ruptures and the RNA comes out. This RNA makes multiple copies of this, and they become wrapped with this kind of shell, and the whole thing form new viral progeny which bursts the cell and comes out. Now, as soon as the cell, this virus entered the body's bloodstream, there are several types of blood cells that can recognize the virus. They are called some antigen presenting cells, some T helper cell, B lymphocytes, cytotoxic T cells, all those things. They encounter this thing. How? First of all, these cells are all part of white blood corpuscles. We learned in our basic biology that we have 3 types of blood cells in the body: red blood corpuscles, white blood corpuscles, and platelets. So these are all different categories of white blood corpuscles. The first cell that sees the virus is called, they are called APC, antigen presenting cell. What do? They engulf this virus, they eat up this virus, and after eating up, they chew these proteins into small fragments and then protrude these small fragments outside the cell surface. It is like showing somebody that I have engulfed this virus which has these proteins. Whom does it signals to? It signals to the T helper cell. So the T helper cell immediately comes to know that this is a peptide that this APC cell, his friend, has digested, and he needs to remember this peptide to identify this culprit. Now, in turn, these T helper cells, they signal the same thing to 2 different groups, B cells and the cytotoxic T cell. The cytotoxic T cell, what do they do is, they come to know that this is the peptide I need to look for, and as soon as it finds out that a cell is infected with this virus which has these proteins outside, it kills this cell along with the viruses. What does the B cells do? These B cells, they are learned to produce a different kind of protein called antibodies. These antibodies are very specific to these spikes and it won't bind to anything else. So these antibodies bind to this spike protein, and there is a pathway it follows by which it kills the virus. Interesting part is, after this part is done, this is transient part, immediate part. After this immediate part is done, these two cells makes their small subpopulation which are known as memory B cells or memory T cells that are stored in the body, who has previously seen this virus, these spikes, and they know that whenever I see this virus in a later period, I have to kill them. That is called immunity. Now, this pathway of creating memory cell is actually is the foundation of creating vaccine. So in vaccine, what do? We create somehow an artificial form of this thing, which will come later, artificially form of this thing, and gets the body ready to know that this type of virus is going to attack sometimes later and I have to kill it. Let's go to the next slide. There are different types of vaccines available and under development. They are called virus-mediated vaccine, which are two kinds, inactivated, weakened. Viral vector, I'm going to talk about all this thing later, don't bother. Viral vector-mediated, which can be again two types, replicating, non-replicating. There can be nucleic acid-mediated, DNA and RNA, two types. Protein-based, there are two types also, protein subunit and virus-like particle. You can see there are different companies and different groups they are working. If you want, you can pause and get an estimate of how many companies are working on which kind of approach. I will try to explain each of them separately. First is virus-mediated vaccines. At least 7 teams presently are working on this. And this is a very established pathway. Previously, they used this kind of vaccine technology for creating measles vaccine and polio vaccine, but it requires extensive safety tox testing. Why? I'll come to that. See, there are two ways. The first thing is you have to insert the virus inside the body. Now the virus is the culprit. How can we insert it into the body? It will cause the disease. To overcome that, there are two ways. Either we weaken the virus or we inactivate the virus. To weaken the virus, what we do is we use this virus to infect some human cells in laboratory, not in human body. So those are human cells, and in laboratory setting, in completely petri dish mediated. So once they infect those human cells in the petri dish, they multiply, they form millions of these virion particles. We isolate them and then again infect another set of fresh human cells and again collect the new virions, again infect another one. We do this till they pick up a mutation that make them less virulent. Virulent means virulence is the ability of attack another healthy cell, so host cell. So we make them less active. So that is the weakened virus. Probably you have heard of Serum Institute of India, who are making the vaccine in the coronavirus vaccine in India, in Pune. So they have used this weakened virus strategy, but they have used a genetic code alteration. That means creating a mutation so that the virals are weakened. We can also inactivate the virus by using formaldehyde or heat. So when we are talking about weakening the virus, that means we have to make sure that the virus is completely weakened and it had lost its capacity to infect further healthy cells. That is the safety part over here, extensive safety testing. Second one is viral vector based. That means we will use a virus which is not a coronavirus, but we will put some part of coronavirus, these proteins, the gene part, the coding part for this. We know that this RNA, they code for different proteins. So we will find out which part of RNA, let's say this part of the RNA codes for the spike. I have another discussion in the YouTube where it's named as bad news wrapped in a protein. You can look at that too. You can get a much better idea. So this small part we clone in another gene. That means we insert this DNA, this small gene, RNA gene here, in another viral gene. Which viral? Most of them, they are weakened measles virus or in an adenovirus. Now, weakened measles virus, they are already weakened. They are not going to cause measles in body. So we insert this spike gene there, and then we insert the virus into the body. So in the body, this gene will be expressed, and those spike proteins will be present in the body, but not the whole coronavirus. So part of coronavirus is expressed there, and again this APC cell will recognize those and signal further to create the immune response. Similarly, we can use an adenovirus also, and about 25 groups, they are working on viral vector vaccines. So I'll go to the next one, which is nucleic acid vaccine. As you know that we can insert a DNA in a viral vector to make it work. So we can, why don't you use that DNA as such? Yes, we can. So we take a DNA and we insert this coronavirus spike gene in there and inject the DNA directly in the body, directly in the human cell. Then this part will create the spike protein. Those spike protein in turn will be recognized by the antigen presenting cell and elicit further immune response. Similar is the case of RNA vaccine. Instead of DNA, we put the RNA. That RNA will directly create the protein. So the difference is, if you inject a DNA, there is an intermediate step called transcription. The DNA will create another nucleic acid, which is called messenger RNA, as known as mRNA. That will give the spike protein. This process is called translation. But for RNA, this step is skipped. We can go directly for translation, and the proteins are released. Now the eventual thing is we need to insert some protein. So the question is, why can't we use protein directly? Of course we can. There are vaccines approach where directly spike proteins are used. For coronavirus, spike protein and M protein, that's also another protein on the envelope of the virus, on the shell of the virus. And we inject that in the human body. So again, the APC will see these cells and go for further immune response. We can use a virus-like particle. That means this is almost the same as the coronavirus, but artificially created. And there is no DNA or RNA or anything, any kind of nucleic acid inside. So the virus will not multiply inside the body. Only it will stay and act as a mimic of the regular virus, a live coronavirus. But it is not a live coronavirus. It cannot replicate. It cannot multiply. So when you inject that, these particles, these spikes proteins will be recognized by, again, APC cell, and the basic method is always the same. Now, what are the challenges for developing virus? Number one is safety and toxicity. No government will allow any medicine, any vaccine, any prophylactic or any curative method that will harm the people of the state. So no country will allow that, and for that, safety and toxicity is a very big concern, and people, whoever making this vaccine, whoever is developing this vaccine, they have to be very careful about safety and toxicity study. Second is, we most of the time recently, what you have already heard that the Serum Institute of Vaccine, they started producing the vaccine before getting any result from human. So it is not sure whether the vaccine will work on human. We know that the vaccine works for animal, but there is no guarantee that it will work in human. Some guarantee is there, but not 100% guarantee. Second is, what is the immune response does it need to prevent coronavirus? Nobody knows. How many molecules of that antibody, that blue thing that I discussed over here, here. How many of these proteins need to be there to create this response? Nobody knows. Second is, how long the vaccine will be effective inside the body. That means some vaccines are longer duration, some vaccines are very short duration. We need a longer duration vaccine which can stay at least for a year or so. So we don't have to take this vaccination every 2 months or so. So these three are the main challenges. Now, lots of people are working on that. In America, there are a huge group working on that. Some of them are public and non-profit, some of them are academic, and some of them are private and industrial. Private and industrial is actually eating the big chunk here. So more than 70 vaccines are under development. 3 are under human testing. What is the situation in India? In India, the situation is 6 companies are working. Zydus Cadila, Serum Institute of Vaccine, Bharat Biologics, Biologicals E, Indian Immunologicals, and Minvax. Of which I got some information from Zydus Cadila and Serum Institute of Vaccine. Zydus Cadila is using 2 approaches. One is the viral vector. You can go back and check what viral vector is, or a DNA vaccine. The status of viral vector is, it is in animal testing, and the DNA vaccine is ready for clinical trial. Expected date of launch is not yet declared. Serum Institute of Vaccine is working with a weakened virus in collaboration with Oxford University and Codegenetics. It is in the production level, but they are hoping that the clinical trial will be successful. If everything goes well, then October 2020 is the expected date of launch. Bharat Biotech working on a viral vector. That much information I could find from internet. Other informations are not available, and nothing is available from other. Only thing that Minvax is, so far I know, is a part of is a collaboration of Indian Institute of Science in Bangalore. This information I got from the Pharmaceutical Journal, which was published in March 2020. So I think that's it. Thank you very much.